Alright, here I have completed my, let's just call it the separation of my long sequence into the shorter sequences. In other words, I have taken my non-coding region that we found in the last uh, video, and I have put it on its own here, called it non-coding sequence before LacI, and it goes on, at least on this FASTA file. So you need to associate this with a particular FASTA file and say that this is from 1 to 78, and here is the sequence. GAC, GAC, AAT, AAT. Okay, it looks good. And then I have my coding sequence here, and we identified that in the last video using Regulon DB to download the open reading frame, and we found that between the green and the red, and that is the LacI open reading frame. Let's make sure we have this right. GTG, GTG, yes, okay. And then we end over here with TGA, so we go down here to the bottom, and TGA. All right, that's right here, so we're good. All right, then we go on, and so you can go and check this, and of course you can find the location on this sequence using my app here. If you put yourself anywhere after a letter, you see these numbers down here change, and that's the location. So if I put my cursor right after the C down here at the bottom, it's telling me that this entire sequence is 7,477 nucleotides. All right, and you can go place it anywhere, and it tells you where you are in that sequence. And I found the locations, and I put them into this document. So I now have these separated so that I can look at them individually and find the things and the locations where I know they are at. So let's go take a look at our let's see, at our at the content here on D2L, and let's look at the project instructions again. All right, and let's see, well, that's actually not what I'm looking for. I want the how to bacterial annotation new 2023. All right, so the one thing we can look for are Shine-Delgarnos, which we've already kind of gone over that. But realize what we said was that Shine-Delgarnos are AG rich, and they are what, 3 to 13 upstream from the start codon. Well, we now have start codons that we are reliably sure about. We maybe weren't so sure before, but now we are. So I can go back and find the Shine-Delgarnos in the non-coding sequence, because realize this is where the ribosome binds. So to code that, or to read that open reading frame, it has to be before it, and it needs to be about 3 to 13 before that. So if I'm going to do this, here is my LacI, so this would be like 1, and it has to be 3 to 13 before that. So how far before? Now here's 3, all right, and 3 to 13, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13. So there needs to be a Shine-Delgarno sequence somewhere right about here, right before, what does it say in the form? It says it needs to before the start codon. So let's look for this 3 to 13 before the start codon. Doesn't mean it can't be outside this a little, but generally this is structurally where it needs to be for this to work. So we are looking for A's and G's. All right, now it's looking for it in the RNA because ribosome will be attaching, and we are in the RNA, so we're good. We have to make sure we recognize where we are at all times. So let's take a look, and we can just look at it in our new thing. So where I have it grayed here, this should be where we find our Shine-Delgarno sequence, and it'll be in a place where we have lots of G's and A's. And it has to be 13 before, 3 to 13. So we rule out this TAA. That's -1, -2, -3. So it can start right about here and go backwards. And where do we see a bunch of G's and A's? Well, this entire region is mostly G's and A's. Yes, there are some there are some T's in here, but this region is mostly G's and A's. Here are two T's. I would say for sure that this is a Shine-Delgarno. Now, I would say this also makes for a good Shine-Delgarno, but this one is in the right location. So you might give me two options. You'll go, well, if this piece is in the right location, this piece looks like a better one because there are fewer T's. There's just a TC here. Maybe knock that TC out. And this looks a little better right here. All right, but this one is in the right location. So either one, you could say, let's go ahead and do this. Let's put some color on this, maybe, to make this kind of interesting. So what color can we do? We'll make one of them, I'll just make this one orange. And we'll make this other one, we'll make it a different color. We'll make it purple. All right, and what would you do? Well, you might tell me, all right, somehow after this, purple, you know, looks better in terms of exact sequence. In other words, G's and A's. And not these exact words, you might say it more eloquently here, but orange, you know, put the location in there too. Remember, this would be what, 78, 77, 76, so that would be 75 something to 75. So 1, 2, 3, 4, 5, 6, 7, 8, 9. 67 to 75 is equal to orange. You might throw that in, you know, like this. 65 to 79 or something, whatever it was. 65, 66, 67, 68, 69, 70, 71, 70. Okay, 70. Oh, 70. Let's see. That's 78. I guess it's 70. What was it? 69. 2, 3, 9. Oh, okay, 66. There we go. Something like that. But orange is in the correct location. So you can give me both of those, and then I would say you'd probably have to go with orange because it is in the location. It's good enough. Yes, there are a couple of T's in there, but if you'll notice the explanation, if you go back to our explanation, the consensus sequence here, but it's just AG rich. It doesn't say it has to be pure. It doesn't have to be all AGs, but it does say it needs to be there, all right, because of the structural limitations of the ribosome. It needs to be within 30 base pair. So 30 base pair, let's count back 30 and see how far. Let's go back. And so this is 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23, 24, 25, 26, 27, 28, 29, 30. So you know what? Both of them are legitimate. Both of them are probably functional Shine-Delgarno sequences. So maybe you can load up on 2 ribosomes ready to go right by each other. I don't know if that's possible, but definitely the right answer would be to put both of them in because they are within the 30, and this one is better for location, this one is better because of the sequence. Now, what is the exact sequence? The best sequence is AGG, AGG. Do either of them have that exact sequence? So let's go look. AGG. No, not quite. It's AG. So this one isn't quite the sequence. This has an AGG, G, and then a T. So this one is not quite as good a sequence as this one. Let's see what other sequences we have listed as commonly AGGA or GAGG. So let's try AGGA, see if that is here. AGGA. No, we don't have AGGA. All right, so nope. But how about what was the last one? GAGG. Do we have that one in there? Oops. Where am I here? AGGA. Nope, don't have that one either. So we don't have a definitive location for Shine-Delgarno's either one, but both of these meet the criteria, so you could say there are potentially two here. You would have to do laboratory experiments to find out which one, if not both, work as Shine-Delgarno sequences. I suspect they both are functional Shine-Delgarno sequences. This one has a better sequence that fits the parameters, but this one is a little closer. Maybe it's too close. It says 3 to 13, but sometimes the like I said, either one could be a Shine-Delgarno sequence. Probably should include that G right there. There we go. All right, but that's a Shine-Delgarno sequence, and we can. That would be how I would expect you to write this in. Now, let's go and look at the next requirement for this. We go start codons, and so you need to identify the start codons and the reading frame, which we technically have already done, including the stop codons. So technically we have already done that in this one. This one is a start, so let's make that one green. All right. And then the stop codon, let's make that one red. All right, stop codon. And then the non-coding sequence in between. Now, this is the between the I and the LAC-Z open reading frames. So if you remember the way the LacOperon is set up, there should be a whole bunch of things in this sequence here. All right. There should be like a tata, so we need to find that. So let's okay, purple. Oh, I didn't label this. So in this non-coding region, let's say Shine-Delgarno. Let's undo this. There we go. Shine-Delgarno. And maybe I'll do something like this to show purple. Maybe I do this for orange. You can do this anyway. I'm just saying that this is just to make it easier to read, to know what we're talking about. So our Shine-Delgarnos, you might put in those locations. I won't do that here. Alright, just that kind of thing to kind of put it, some narrative, something to explain what's going on and where the Shine-Delgarno is, and maybe give me some explanation. Might give me some explanation. Here's your start, and maybe explain that this is a non-standard start codon, and this is a standard open stop codon. And give me your locations, so you might add those things in. So now we go to, we're not ready for termination yet. Let's go and find more Shine-Delgarno. There might be some more interesting things to find in the Shine-Delgarnos and the others. So let's go see, where should we find Shine-Delgarnos in this? Well, we can find shine Delgarnos in any of the non-open reading frames. So we need to go and find more shine Delgarnos here. So this is the non-coding region. So we need to find it within 30 bases. So let's count back 30 bases. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23, 26, 27, 28, 29, 30. I'll go back a couple more just to do it. So we want to find our shine Delgarno somewhere in this region, somewhere around here. So we want, what's the definition? Let's go read it again in our explanation. Shine Delgarnos are AG rich. Doesn't mean that you can't have other letters in there. These are the consensus. You need, structures like this. Look for them about 3 to 13 upstream, but they have to be within 30. Okay, so we found our 30 before the start codon, and we're going to look strongly in the 3 to 13. We're also going to look for just a bunch of A's and G's, where most everything are A's and G's. If we can find the consensus sequence or one of these commonly found sequences, we're going to say, oh, hey, we think that's it, but not necessarily. All right, it's just there. So here is GAG, and here's some GGA. Hey, that's AG rich. So that one might be good. Let's change that color to yellow. All right, and where's another AG rich? Here's an AG rich region. We'll count that. So there's another AG rich region there. This is the orange here, is the region where you could find it, but no, those are our two best candidates for shine Delgarno. Where is the 3 to 13? So 3 to 13 starts here. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Somewhere in here. So prime location is in here, and sure enough, AGGAA is in here. And is that one of the prime ones? Let's go back and look. AGGA. And so I'd like to find AGG, but AGGA is one of the most commonly found. So let's go see if that's there. AGGA, and yes, there is an AGGA there. So this one's a little bit more, it's in the 3 to 13, which is the most likely. I would say that this right here, I'm going to make that a different color, really consolidates our thought that, yeah, that's a shine Delgarno for sure. Not as sure which one works up here, right? But because of the AGGA, yeah, we don't get AGG, but AGGA was one of the more common ones found. So I'm pretty sure that that is a shine Delgarno. Of course, here we have our standard start codon, so we'll make that green. And it's long. Here's our stop. We'll come back maybe and find some other things later, but for now, that's all we need. We're going to find some more shine Delgarno in here. So, easy to see there. All right, so there's our common piece, and so yeah, that's past 3, yeah. And then AG, not so much. AG, there's some AGG back here. I don't like any of the other parts along here for shine Delgarno. I think that's the only one in that part. All right, now let's go for the next non-coding part. AGG, up right there. Here, we can include that T in there. That's pretty good. Yeah, I like that one, and I don't think here's A. Now, I don't like any of the others. Maybe back here I do, but that's too far, I think. So let's see the 30. 1, 2, 3, 4, 5, 6, 7, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23, 24, 25, 26, 27, 28. Nope, nope, gotta be right here. This is our choice here. Non-coding after LacA, no, we don't have anything we shouldn't be looking for Shine-Delgarnos after LacA. So we found our Shine-Delgarnos in these sequences. Found our start and stop codons too. So we kind of have a location. Let's go ahead and mark our start and stops. So here is our green one for start. And here's our stop, UAA. Yep, that's a stop. Okay. And then we have, oh, look at here. Is this another, looks like another odd start. I don't know about LacA. If it is, there's an ATG right here, but this kind of indicates that maybe we have a non-standard. Let's look at that requirement. TTG, is that a possible start? TTG is, so this might be another non-standard start to be on the lookout for. And here's a standard stop. There we go. So here we've made a good start. We've got like the first two parts of our annotation done. All right, we have our, at least for the LacOperon here, I have found the Shine-Delgarno sequences and I've found the start and stop codons. So that means the reading frames. Now, termination, now that's a different, we will need other things to find termination, and that's going to come at the very end. I'm going to make a whole video like that. What we can do on this one is look for promoters. So I'm very quickly going to look for promoters. I may need to do a whole video on this, but let's see if we can find a Tata box. If we don't find a Tata box, then that means we need to go and look for other promoters. All right, and so, but I think I can get away with doing Tata boxes, which is the most common promoter by far in an E. coli. So we have the consensus, and now it says to be about 10 base pairs before transcription start. So transcription start, we really don't know where transcription start is, but essentially this is going to be very early in the sequence. All right, so let's we don't know, it's really hard, but we should also find this other sequence at minus 35. So we have minus 35 and minus 10, so we really need to be looking for both of these. This is the Tata, but you really need this as well. And the optimal spacing is at minus 35 and minus 10. And we know that it has to be before transcription start, which is going to be before the Shine-Delgarno. So, but everything is, it's going to be tricky here to figure it out. But we need to look at least minus 10 and minus 35 for something that looks like a Tata box. Now, a Tata box doesn't really mean a TAT or a TAT-AAT. This is just the most likely consensus, not necessarily that you just need AT-rich. So we're going to be looking for AT-rich regions that are more or less before the Shine-Delgarnos. So we know these should come before, well, they could be anywhere, but yeah, it should come before the Shine-Delgarno. That's all we really can say. So we need to look for AT-rich regions before our chosen Shine-Delgarnos. So let's go look at our regions real quick. And so we're looking for AT-rich regions before the Shine-Delgarnos. Let's go way up here and do the first one. And let's see, we need to come up with a color scheme for Tata boxes. Let's make this a different color. Let's make them blue. Let's make them a dark blue. You can do whatever you want, but AT-rich region. Well, share our shine Delgarnos that we think exist. So look at that. There's a G in the middle of it, but that might be okay. Does not mean that it has to have that. So AT-rich, I don't think TAT is enough. I think you're going to have to have a stronger region. Would this count? It is an AT-rich region, but it's not staggered TAs. I don't think this one would count. However, this one, man, you got 2 letters in between there, but you do have an AT. It's a possibility, but less likely. Now, why did I like this one? Well, it's got ATs, all right. It's got A's staggered by T's, and this one here, and only 1 G in the middle. I kind of like that one. This one has 2 letters between your AT-rich region. I like that one much less. I would say this one is the more likely one. This just isn't enough A's and T's, and this is it's not AT. It's really A's and then T's. It's not A's and T's mixed very much, so I wouldn't count this. But these 2 regions are likely Tata boxes. Okay? So that's how you do that. Let's go do some more examples down in the next one. Now, where else are we going to find a Tata box? We're going to find one before LacZ. So that means this is LacZ, so we should find Tatas somewhere in here. Oh, well, we also need to go back. I'm sorry, we're not done up here. We need to find it in a specific region, and then we need to find that second piece. So, hey, we are not done yet. So where does it say in our paper here? It needs to be at -10 from transcription start. And transcription start must be at least 1 letter before your shine Delgarno. So let's take a look at that again. So maybe we can find some more. So if this is a shine Delgarno, then we need to be at least before this one. And that one is before it, but we need to be about -10. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Yeah, that's about right. -10 or -11, that would be about right here if shine Delgarno, if transcription start was right here. If it's here, let's say if it's somewhere in this region and we're using this shine Delgarno, we could have some more. So let's say 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. That isn't quite right. I'm kind of liking this first one. Because this one is just too far out to be the Tata box if we're going to have these restrictions. So of these 2, we do have something to say. We can say, because if this is the shine Delgarno and transcription start is somewhere along here, we're good. Now, could transcription start site be somewhere downstream? Yes, it could. We'll get to that later. But I think this makes a better case for this one, assuming it doesn't rule it out, because the -10 should start right there. So in other words, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. In here, those C's, one of these C's might be the first letters of the transcription start site. But anywhere on here, you can still leave these 2, but bacteria tend to conserve space, and so they're not going to have a lot of letters before the shine Delgarno, before transcription starts. It usually starts. So I just happen to know that. So I know transcription start is going to be very quick here. So having a Tata here would fit, would be fine. This seems like a lot of wasted space, and bacteria don't like to waste space, so I don't think this is a shine. I would say this is probably your Tata box, and it looks better than this one. There are 2 letters in between that one. I don't like that. Only 1 G, that's fine. Okay, so this one could be a Tata box if this is located for this one. And there's nothing keeping you having from 2. This could be one that's degenerated and another one is cropped up to make it work. So this could be the Tata box if transcription starts here, and this could be a Tata box if transcription starts like right here. And you could explain all of that, that you have a choice here. It could be one or the other or both. Don't think this one is, and I didn't think this one is, this one was because you don't have staggered A's and T's. So of these, I think this one is the best choice. All right, that one right there is the best choice, and this would probably be your best choice for the for your Shindell Garno as well. So these two seem to be going. Now, if this is your -10 site, that means there's also a -30. If you look up here, remember we were saying that there's also a -35 sequence, the TTGACA. So TT, two T's and then GACA at -35. So if we say that blue one was the -10, let's go back and see what's 25 letters before it and see if we can find that. So 25 letters before that would be 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 18, 19, 20, 21, 22, 23, 24, 25. All right, so somewhere right here where at the end of our blue and right here before these GGs, we should find somewhere in that region that sequence we were just talking about, which is TTGACA. TTGACA. So TT. All right, we don't have two T's anywhere in here, but we do have a T. TTGC. TTGGCG. It's close, I guess, close enough. This is not always there, but it is often there. You look for the, if you think this is the Tata, then you look for that somewhere down here. And let's look at it again real quick. All right. TTGACA. Okay. TT. GA. Here's a CA. GACA. It might be there. That might work as one. This is not a. Now, if you find one that's specific, TTGACA. Let's see. Do we see a GACA anywhere? GACA right there. I wonder what two letters come before that. I wonder if those are TTs. Now, if we had a little bit more sequence here and there were two T's here, oh man, we found it. But we don't. And I don't see any GACA anywhere near, but there's a GACA. You can make an argument that, hey, maybe there are two T's before this one, and that's the correct one. You could also make the argument that maybe the end of this blue piece, here's a CA, but we'd need a GA. Yeah, it's hard to say. May not exist here. It is not required to have it, and this one is much less specific. It can be much more flexible. It doesn't have to be exactly that, but the location is important on that one. So you could say, hey, maybe that's it. Maybe it's here and we can't see the two T's. But here, maybe that one would work with this. Maybe this really is the Tata box. And then back here, you would have plus the two T's would be the -35 sequence. You could make all kinds of arguments here. This is not without experimental evidence. In other words, going into the lab and testing for this, you really don't know. All right, the bioinformatics is limited, and you can't you can't trust a computer. Humans make much better judgments on these than the computers do. Like, we noticed that it could be here. Computer might not notice that. We can make a judgment that that's kind of close in here, that sort of thing. So human judgment does prevail in bioinformatics at this point. AI is not always smart enough to see the things that we can see. Okay, let's see. What else can we do? I think that's enough. Let's real quick go down and look at the other at this one to see if we can find a Tata box. If if this is our Tata box, then we want about 25 before. So let's row. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Yeah. So our Tata is probably in that group right there. That's about the right space. Here is your Shine-Delgarno here. Now you could have Tata in here, I guess. Here's a TAT. That's not quite enough. Here's some T's. That's pretty good. Hard to say. This is a real good one, actually. That would probably be a really good sequence for a Tata box. But, hard to say. I would think your best bet is probably something in here. Yeah, somewhere in there. That's about right. I guess 1, 2, 3, 4, 5, 6, 7, 8. Yeah, that's about the right spot. If this is the Shine-Delgarno, I think we could make that a pretty good spot. You could say that this is a pretty good spot too. But if this is your Shine-Delgarno, say in here, then your transcription start site might be here. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Well, that would make this your Tata box. So maybe this is the better one for your Tata box. Yeah, right in here. And that would fit with this is the Shine-Delgarno. You don't waste any space. Yep. So let's see what happens at minus 35. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21. So do we see anything that looks like TTGACA? This right here. We're looking at that back in there. TTGACA. So TTGACA, TTGACA. Oh, here's CATT. TTATTGACATT. Maybe here. Minus 35, which would put a Tata box about here. Okay, maybe we are off a little. TTAA. TTGACA. No, I don't see one. Maybe here. TTGACA. That would be my best bet for a minus 35. And that would put your Tata box maybe here. And then your Shine-Delgarno would be way up here. Don't like that. I don't see much indication of a minus 35 here. And you just have to say that sometimes. Nothing particularly looks good. All right. So Tata could be, if this is it, this could be your Tata. This whole region could be like the Tata. You have an extra C in there, but that's high AT rich region. And it is about minus 10. So the beginning of it is 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Oh, yeah. If you started here. One thing to realize, Tata boxes. And I haven't said that, but technically Tata boxes are in the DNA. So really, instead of that sequence we're looking at, they're really just AT rich. But if you think about it, if they're in the DNA, really, what should we be looking for? Instead, we should be looking for ATATTA. All right, so at least for these two, because these two are supposed to be in the DNA. So let's see. Oh, no. It's giving us 5' and 3'. So let's see. If it's 5', you read it from what? We're in RNA, so we are in RNA, so that's fine. But you would still find the complement because they're DNA, so in this one it's ATAA. You can reverse it, it doesn't matter. Um, so let's see, TTs. So at least with these, you can go backwards and they still work. You can also, Tata boxes will work on the complement as well. So it can be on either strand and going in either direction. So kind of think of it this way: A's and T's are really you shouldn't go by this standard here. You should just realize it's an AT rich region. This one is a lot harder to figure out. And maybe if it goes backwards, in fact, let's look for this one going backwards and see if we see ACAG maybe somewhere. And because it will work, these two will work going in the opposite direction on the other strand even. So if we can find something similar to those, even if it's going in the other direction, that would be a legitimate choice. They don't tell you that in, and I didn't tell you that either, I'm sorry. Let me go look at this. So can we find this going kind of in the opposite direction? AG or TGC, TG. Maybe. Let's go back up to that other one real quick and let me look at it again. We're looking for something here, so TT. Still don't find anything good up here, really. I don't see just this one. Yeah, it just doesn't look very good up here for that minus 35, unless it's something way back here, and that's a little far. But that's probably the hardest one we've done, is to find that minus 35 sequence. That minus 35 sequence is kind of tricky. I would take all kinds of arguments if you want to make an argument for one. The only real way to find it is experimentally in the laboratory. All right, I think that's enough for this. I think you get the idea for finding these few things that are in the non-coding region. Let's go look and see what's up next. This is promoters. I just wanted to look at Tata boxes in particular just to give you an idea. Read the instructions and try to find it. Listen to what I say, listen to what it has here, and kind of mix that together. We'll do terminators at a later time. The reading frame, certainly, we pretty much got that down. If you have the amino acid sequence, you can get the reading frame. And that means start and stop codons. And then close to that, I think you see how to do Shine-Delgarno. Tata boxes, maybe we'll do a whole another one on just promoters, and I'll go over Tata boxes again. There's a lot of information here. We should find mainly Tata boxes unless your genome, your operon has something to do with these. If your operon has something to do with flagella, heat shock, or nitrogen starvation, you shouldn't find these promoters. Just in general, you should find the Tata and the minus 35. So these are the sequences you find the equivalent and their locations for Tata and the minus 35 in between these. So 16 to 19 between these. So this one is minus 10 and this one is like minus 35. So minus 35 is where it starts. Minus 10, so T should be at minus 10 if it's perfect. This T should be minus 35 if it's perfect. It's the way you think of it. And then put 16 to 19 in between them. Here you would put 13 to 15 between them. That sort of thing. So minus 10, minus 35. And you should find them about the same location. But they're going to be slightly different for that. And we'll go over transcription start at a different time. Okay, that's plenty for this video.